Well, good morning, everybody. This is the second and last press briefing of EHA during this meeting. Good that you are here, despite the delay of the train and other complications. My name is Ton Hagenbeek. I'm a hematologist from the Amsterdam University Medical Center. I have the privilege to chair this press briefing with quite some hot topics to be presented to you. Every speaker has 5 minutes. I'm very strict to that. And then we have time for 5 minutes discussion. And I guess you're going to be here after the press briefing if people want to have a personal video with you or whatever. So I stimulate you to come up with questions after each presentation. And I think we should get started with our first presenter all the way from Beijing, Dr. Let me see whether I pronounce it correct, Dr. Ying Tan. It's not that difficult.
Okay.
And she will discuss, why don't you go ahead, on a new anti-CD20 CAR-T immunotherapy that saved the life of children with leukemia. Dr. Ying Tan.
Shall we start? Thank you. Good afternoon. I'm Jing Tan. It's it's the same name, and it's my great honor to stand here to talk about our study about CD22 CAR-T immunotherapy. My title today is a new anti-CD22 CAR-T immunotherapy saved the life of children with leukemia. I'm from Beijing Boren Hospital in China. So I have nothing to disclose.
But you have to wait for another 5 seconds.
Okay. It's a long time.
Yes.
Okay, move on. Acute lymphoblastic leukemia is a fatal blood cell tumor. And it is usually treated by chemotherapy and allogeneic hematopoietic stem cell transplantation. In recent studies, CD19 CAR-T immunotherapy have been successfully used in treating refractory or relapsed acute lymphoblastic leukemia. However, some patients have still relapsed, and usually they were resistant to a secondary CAR-T cell infusion. So our purpose is to explore a humanized CD22 CAR-T immunotherapy in pediatric refractory or relapsed acute lymphoblastic leukemia. So we have 15 patients in our study. All of them are children, and all children are refractory to chemotherapy. And 14 children had relapse or no response to previous CD19 CAR-T cell immunotherapy. And 4 children had relapse after allogeneic hematopoietic stem cell transplantation. So in our study, all heavily treated kids with refractory or relapsed BAL will die with current therapies. So here is our protocol. First, we get CAR vector from Shanghai Yake Biotechnology Company, and we connect patients' lymphocytes from their peripheral blood. We do the CAR vector transfection and CAR-T expansion in vitro. After about 1 week for cell culture, we get CD22 CAR-T cells. So we infuse the CAR-T cells into patients, and after that, we monitor and treat the side effects. The major side effect is the cytokine release syndrome. And after that, we do the evaluation on therapeutic efficacy. So the cell culture and the clinical use are both in our hospital. Here is our results. First, we get high efficacy in our study. The complete remission rate is 18%, and the partial remission rate is 6.7%. So the overall response rate is 86.7%. We also evaluate the long-term outcome. We had 13 response cases, and we had progression-free survival rate is 91.7%. On the other hand, we think this therapy is quite safe. This picture showed major side effects, cytokine release syndrome in all patients. We find no mild CRS in all patients, and nobody died from our study. And we can see the neurotoxicity, and there's no severe neurotoxicity has been observed. And we found no difference between the non-HCT patients and HCT patients in CIS. So here is our conclusions. CD22 CAR-T immunotherapy brings a hope for patients with refractory or relapsed BAL who even fail to CD19 CAR-T immunotherapy. And we think it's is quite safe. No children die, and no children has severe side effect during the study, and even if post-HCT patients. So if anyone wants to know the details of our study, welcome to our presentation. Thank you.
Thank you very much. Thank you very much for this clear presentation, and there's time for discussion now, please. Yes, the microphone. And your affiliation, please.
Hi, I'm Neil Osterweiler with Hematology News. So why do you think that the CD22 target worked where the CD19 target did not?
Because both the CD22 CAR-T immunotherapy and the CD19 CAR-T immunotherapy can work. But in our study, all the patients had received previous CD19 CAR-T immunotherapy, so they didn't. So they had resistance to a secondary CD19 CAR-T infusion. So we used the CD22. And
were there any cases? How long have they been followed up?
In this abstract, it's about the patient had been followed up for about 200 days, but actually, in most patients had a free survival until now.
And did you have any cases of loss of the antigen?
Lost the CD22?
Yes.
The density of CD22 expression is decreased in some patients when relapse.
Thank you.
Further questions? Maybe I may ask a question?
Yes.
I mean, these patients were full with leukemia, right, at the time that they entered your study, and you did not see any significant cytokine release syndrome symptoms, because it was a very safe treatment.
Yeah.
So how do you explain that? I mean, does it have to do with the construct, with the typical construct, that you did not see cytokine release syndrome?
So you mean when the patient had a cytokine release syndrome, how we treat?
No, I mean you presented the slide where you showed that there was virtually no cytokine release syndrome. Does it have to do with the construct? What do you think? Because they were full with leukemia, would expect some patients to have cytokine release syndrome.
Sometimes I'm very interested in this symptom. So we think maybe it's the density of the CD22 expression on the leukemia cell, or the affinity of the CD22 antibody against the antigen. So we think the grade of cells is lower than the CD19 CAR-T immunotherapy. When the patient has this high leukemia burden, when we do the CD19 CAR-T immunotherapy, it's not the same CIS.
Okay. Further questions? Then again, yes, please. The microphone.
Nicola Sigmund Schulze. I'm here for the German publication for physicians, Deutsches Ärzteblatt. I have a question. Do you have any data about persistence of these CD22 CAR-T cells? How long do they persist? Do you have data about that?
I had data on the persistent, so I will show in my oral presentation. So usually the CD22 CAR-T cells persistent for about 1 month by flow cytometry. We didn't do the gene sequencing on the patients, so we think it's the same as our CD19 immunotherapy, and the longest one is we had detected is about 3 months. Thank you.
About 3 months.
Yeah, by flow cytometry, not the sequencing.
Further questions from the press? Yes, please, in the back.
Hi, Nancy Melville with Medscape Medical News. I just wanted to ask about what was the average time to failure on the CD19 CAR T?
Could you pardon?
The average time duration to fail on the CD19?
Yeah. You mean how long when the patient...
how long did it take? Yes.
I mean the time between CD19 CAR and a relapse after that?
Yes.
Different patients had a different relapse time. The shortest one is about 2 months after CD19 CAR T immunotherapy.
Okay.
But all the patients in our study they didn't receive the CD19 CAR T immunotherapy in our hospital. So many people from the other hospital and they received the CD19 CAR T immunotherapy in other hospital.
Okay.
Did you check for minimal residual disease?
Yeah, of course.
And were they all MRD negative?
I will show the data in our presentation. Most of them can achieve the molecular complete remission.
How many, which fraction of the 15 patients?
15, about 11 patients.
11 out of 15 MRD negative?
Yeah.
Okay. And did you consider to go on with an allogeneic transplant in some
of
selected
course.
patients? Did you do that?
Yeah, of course.
So the curve that you showed includes after allo transplant?
Yeah, so we got a high survival rate.
How many patients did you transplant?
In this abstract, 6 patients have received allo transplant.
Okay, okay. Okay, well, thank you very much again.
Thank you.
And we proceed to Dr. Meletios Dimopoulos, who has, from Athens, Greece, who has done an excellent study, the INNOVATE study, dealing with ibrutinib plus rituximab for patients with Waldenström's macroglobulinemia. Dr. Dimopoulos.
I have to get to my slides.
Does it work? Yeah, you did a great job. Yeah.
Oh, but
you were not nervous? Were you a bit nervous? I'm always nervous.
Good morning. We are changing gears to Waldenström's macroglobulinemia, which is a relatively infrequent disease. Its incidence is about 10% that of myeloma. It is a disease of the elderly. And in this particular study, we investigated the activity of rituximab and ibrutinib in patients with macroglobulinemia. These are my disclosures. We know that Waldenström's macroglobulinemia is a low-grade lymphoma characterized by the secretion of monoclonal IgM and also by lymphoplasmacytic neoplastic cells that infiltrate the bone marrow, the lymph nodes, the spleen, and sometimes they have also extra-lymphatic invasion. A few years ago, it was described that a gene, MYD88, had a specific mutation, and this was present in approximately 90% of patients with Waldenström's macroglobulinemia. And also we say that we know that in these patients there is a constitutive activation on NF-κB pathway via Bruton kinase, tyrosine kinase. This led to the exploration of BTK inhibitors, and the first one was ibrutinib in the treatment of this disease. Indeed, ibrutinib was administered in the context of 2 phase 2 studies, and it showed responses up to 90% in patients with Waldenström's macroglobulinemia, and this led to its approval in the United States and in Europe, and this is the first drug actually approved for the treatment of this disease. In this trial, we wanted to investigate whether the addition of ibrutinib to rituximab may improve the results of these patients. This is the design of the study. We included 150 patients, some of them previously treated, but also later in the study, we allowed newly diagnosed symptomatic patients to be included. regarding previously treated patients. They could have received rituximab, but they should have not been resistant to rituximab, and the last rituximab should have been given more than 12 months from study entry. The patients were randomized on a one-to-one basis, and they were stratified according to IPSS, to number of prior regimens, and to ECOG status. In both arms, rituximab was given at a standard dose, but at an extended fashion on weeks 1 to 4 and 17 to 20, because we and others have shown previously that this is more active in Waldenström's. In arm B, patients received oral placebo. In arm A, patients received ibrutinib at a standard dose of 420 mg once daily until evidence of disease progression. This is the main finding of the study. The primary endpoint was progression-free survival, and as you can appreciate, with a median follow-up of 26.5 months, there is a very significant improvement of PFS in favor of the combination of ibrutinib and rituximab. The median PFS in the arm B with ibrutinib and placebo, with rituximab and placebo, was 20.3 months, whereas it has not been reached in the investigational arm with a hazard ratio of 0.2. Even more significant was the observation that the combination of ibrutinib and rituximab was able to improve the outcomes of patients that had specific genotypes. Previous experience with ibrutinib has shown that when we have patients with wild-type MYD88 or when we have patients with mutations at CXCR4 gene, the response to ibrutinib and the PFS is shorter as compared to patients without these features. In this particular study, we were able to show that the combination of ibrutinib and rituximab essentially abrogated these adverse events. Furthermore, the safety profile of the combination was similar to that of rituximab, with the exception of a few cases of hypertension and atrial fibrillation, which are known side effects that occur in a minority of patients treated with ibrutinib. We conclude that the combination of ibrutinib and rituximab improved progression-free survival regardless of prognostic or genotypic factors. There was a significantly higher response rate with ibrutinib and rituximab versus placebo and rituximab. Furthermore, in 96% of the patients who presented with a hemoglobin less than 11, there was a significant improvement of the hemoglobin after treatment, which is a very significant parameter for patients' quality of life. There was no IgM flare that we see with rituximab, and the discontinuation rate due to adverse events was low, around 5%. We believe that this combination now could be considered as one of the standard of care for patients with both newly diagnosed or previously treated with Waldenström's macroglobulinemia. Thank you very much.
Thank you for presenting this. It's really a major step forward, I would say, in the treatment of Waldenström's disease. Questions from the audience, please. Raise your hand.
Hi, Neil Osterwald with Hematology News. Anything new here from what you presented at ASCO?
No, we present this study was presented at ASCO a few weeks ago, and actually it is published online in the New England Journal of Medicine. And we thought that it would be a good opportunity for our European colleagues to hear this data this afternoon, because, as you know, many attendees of EHA, they don't go to ASCO from Europe and also from other parts of the world.
Okay, thank you.
Thank you.
You concluded that this might be one of the new standards of care, or would you rather say this is the new standard of care?
You're right, Professor Hagenbeek. However, I believe that there is still room for improvement in Waldenstrom's. First of all, we would like to develop regimens that may deepen the response and clearing the bone marrow. As you know very well, since you are an expert on low-grade lymphomas, these BTK inhibitors do not really clear the bone marrow. The other important question is, especially for indolent diseases where the patient have a very long survival, to develop regimens that will have a fixed duration of treatment. We would like to incorporate a third agent, maybe venetoclax, which has a significant interest in this disease, or calfizomib, because we know that proteasome inhibitors are very active in Waldenstrom's, and actually they have significant activity in patients with CXR, CR4 mutations, to develop maybe a triplet that could be given for a fixed duration of time and to avoid administration of ibrutinib for years in these patients.
Sounds promising. Next question. Anybody? Is it too early to say something about overall survival?
Yes, absolutely. It is too early. And also in this study, we offered the option for the patients who progressed on rituximab to receive ibrutinib. So far, 30 patients from the placebo arm have received ibrutinib at the time of progression. I believe that we will need several years for to reach the median PFS with the control arm, and even more years to see whether there will be a difference in the overall survival.
If there are no further questions, thank you again very much.
Thank you.
And we proceed, we are on time, so that's good news. We proceed to the talk by Dr. Laurie Saint from Vancouver, British Columbia Cancer Agency. And the title of her presentation is Answering the Unmet Need in One of the Most Common Types of Lymphoma. It's as simple as ADC. And you should explain that, Laurie.
So ADC, and I apologize for the pun, but we thought we'd liven things up this morning on a Saturday, refers to an antibody-drug conjugate, which I'll be talking about, which is a novel therapy for a particular unmet need in a common population of patients with lymphoma. Oops. These are my disclosures.
Why don't you sing your national anthem in the meantime?
Tempting, but...
It's always, the time doesn't move. Oh, the time does not move, my God. But there's time for the national anthem then. We would have loved to hear the Greek first.
Perfect. So as many of you might realize, diffuse large B-cell lymphoma is a type of aggressive non-Hodgkin lymphoma, and it is the most common type of non-Hodgkin lymphoma. While this is very curable in a large number of patients, despite our best standard upfront therapy, which generally involves immunochemotherapy or R-CHOP for many of these patients, about 40% of patients either won't respond to initial therapy or ultimately relapse despite that initial therapy. As a next-line treatment, many of these patients are considered for high-dose chemotherapy and a stem cell transplant as a potential next curative attempt. Unfortunately, many patients are not eligible for transplant due to age or comorbidities, and the treatment options for these transplant-ineligible patients or patients who relapse after transplant are really limited. Survival is generally quite short, and really the responses to successive therapy actually worsens with multiple relapses. So we certainly do need better treatment options for this group of patients. At this meeting, I'll be presenting a phase 2 trial exploring the use of a novel agent, polatuzumab vedotin. This is a first-in-class anti-CD79B antibody-drug conjugate. CD79B is a protein that's present on the surface of many normal B cells and is typically universally expressed on diffuse large B-cell lymphoma as well as follicular lymphoma. This antibody drug construct is essentially an antibody targeting CD79B, so it specifically seeks out the tumor target. It's attached to a toxin And when it attaches to the tumor cell, it actually gets absorbed into the tumor cell, and the toxin is selectively released into the tumor. So it's a way of specifically delivering the toxin to the tumor cell while trying to protect the normal body cells. So in this phase 2 trial that I'll be presenting, it enrolled patients with relapsed refractory diffuse large B-cell lymphoma, and it was a head-to-head comparison of polatuzumab vedotin in combination with bendamustine and rituximab versus bendamustine and rituximab alone. The primary endpoint of the trial was the CR rate as determined by PET scanning at the end of treatment, and this was significantly higher in patients receiving the polatuzumab vedotin with the BR, so it was 40% versus 15%. And importantly, this translated into a significant improvement in progression-free survival, which was almost tripled with a median progression-free survival of 6.7 months versus 2 months, and also quite remarkably into an improvement in overall survival, which more than doubled, with the median being over 11 months compared to under 5 months. So in this randomized phase 2 trial, it really is the only so far head-to-head comparison of a novel targeted agent against a standard therapy in this patient population that's ineligible for stem cell transplant. And it demonstrated that the combination of polatuzumab vedotin with BR significantly improved the response rates, progression-free survival, as well as overall survival. And based on these encouraging results, this drug has been granted breakthrough therapy designation, both by the US FDA as well as PRIME designation by the EMA, and it will be explored further in other combinations in ongoing studies. Thank you.
Thank you. Thank you very much. Excellent example of targeted treatment. This was done in 80 patients with DLBCL, right?
Correct. So it was 80 patients in total, randomized 1-to-1, so 40 patients in each arm.
Right, right. Questions from the press? Go behind the table.
Great data. Do you believe that bendamustine and rituximab is the appropriate companion in this type of histology with this exciting new drug?
Mm-hmm. So I think that, an excellent question. The control arm, obviously, with bendamustine and rituximab alone, does not do very well, but I think it's fair to say that there's no standard of care in this population and no known, magical drug combination that does actually do very well. Moving forward, I think whether or not this drug will be combined with BR or other novel therapies, I think is an important question. I think highly likely, other combinations will also show, hopefully, that it's beneficial.
Hi, questions?
Yes. Dr. Demopoulos asked my question pretty much, but are there other antibody drug conjugates with different toxins, partnered with the antibodies?
Yeah, so you're probably familiar with rituximab vedotin, which is the antibody drug conjugate that's approved for Hodgkin lymphoma.
Yeah, I meant others in development, sorry.
Correct.
Yeah.
And so there, in terms of non-Hodgkin lymphomas, there are a variety of antibody drug conjugates in development. Some of them are targeting different epitopes as CD22. All of them are really still in very early stages of development, and some of them do have other toxins attached.
Well, the brentuximab vedotin directed against CD30 also may find a place in a subset of DLBCL patients that are CD30 positive, about 20-30%. Mm-hmm. You're running a phase 2 trial on this particular antibody drug conjugate in DLBCL. You didn't mention anything in terms of toxicity, of side effects. Could you briefly summarize? Because there must be side effects, I think.
Yes, so nothing is without side effects. So
right.
In terms of the safety, I think importantly in the combination, there were no unexpected toxicities. So typical to what we'd expect with what's known with this drug alone and the combination of BR alone, the toxicities were, I think, quite anticipated. There was definitely a step up in toxicity. The majority of toxicity experienced with the triple combination was generally low grade, including peripheral neuropathy. probably the highest grade toxicities that were seen related to cytopenias. So there was a higher rate of neutropenia, grade 3 to 5 neutropenia, as well as anemia with the combination, and a slightly higher rate of febrile neutropenia, but it didn't translate into a higher rate of grade 3 to 5 infections. So in general, the step up in toxicity for the most part was lower grade, including the peripheral neuropathy, which there was only one patient that experienced grade 3 or higher peripheral neuropathy. The majority of it was grade 1 or 2 and largely reversible in the majority of patients.
Were there any deaths related to the treatment?
So this is a very high-risk population,
yes,
as you might expect. Many of them, there were deaths on the study, but it was equal in both arms, and many of them were related to progressive disease.
Okay. Further questions? Well, it was perfectly clear. So what is going to be your next step?
So this drug is going to be tested in the frontline setting for diffuse large B-cell lymphoma. So there's now an ongoing trial called POLARIX, where the polatuzumab will be replacing vincristine, one of the standard agents in R-CHOP. So a head-to-head trial of R-CHOP versus the combination without vincristine and the polatuzumab added in. I think that's a very exciting study that's already underway.
And did you note any difference in response rate between primarily refractory patients, which is the highest unmet need group, versus relapsing patients?
Yeah, I think that's a really important question. So we did break down the study group according to whether or not they were primarily refractory or relapsed and how many prior therapies they had. And across the board, the benefits seemed to be the same in all of those subpopulations.
And any difference between GCB, ABC, subtypes of DLBCL, or is the series too small?
You know, based on how this drug works, we didn't anticipate that there'd be any difference in terms of its potential utility for ABC versus the GCB subtype. But looking at that population, there did not seem to be any difference.
Final question from the audience? If not, thank you again very much, Laurie.
Thank you.
And we move to a new phenomenon in the press briefing that is summarizing some EHA topics that are in focus. And the first one is going to be presented by Dr. Maria Seppellini from Milan. And this focus deals with the basics of sickle cell disease to where we are now.
Thank you. Good morning. It's my honor and a privilege to introduce to you some new program of EHA that tackles high-impact issues faced by the medical world today. These are the two programs are focused on immunotherapy and hemoglobinopathies. I will focus on hemoglobinopathies, especially on sickle cell disease. The goals of these two programs are to raise awareness, provide education, further research, and build a network of experts with the goal to improve patient care. This is a special unmet need in Europe where hemoglobinopathies and especially sickle cell disease were not so common. As you know, sickle cell disease is a disease where almost 30 or 40% of the population in Africa is affected. So this has a very important impact. In fact, the reason why EHA focus on this issue is because it's becoming a socio-economical problem. So that's have a significant impact on health care. The income, the new refugees or migrants from Middle East and Africa are definitely changing the incidence of hemoglobinopathies in Europe. So we definitely need to increase the awareness. And the program is including several activities, several actions, starting from education. Education of physician, of healthcare provider, and also actually the social and political environment surrounding this new real problem we are going to face. Second objective will be to harmonize and to implement registries on hemoglobinopathies in general, including thalassemia, but especially focusing on sickle cell disease, supporting and involving the creation of patient community, and what is one of the intervention is to educate, to diagnose early in life this disorder, maybe also setting newborn screening, and of course, when these points are achieved, the implementation of new treatment will be one of the focus. Just to support this program and just to increase the awareness, June 19 is a World Sickle Cell Day, and we hope that the hematology community will join this day. Thank you.
Thank you very much for this overview on new activities. Any questions from the press? I think this is a very good development that we focus on one of the most frequent hematological diseases. Could you comment on what the most promising treatment at the moment is for sickle cell disease?
Well, actually, the standard, we have to say that the important issue in sickle cell disease is the sickle cell crisis, which very often is not recognized. And when it's not recognized, it can be fatal. And that is the main point. And in Europe, the awareness is really very poor. So having said that, the standard of care can be very easy. It's first of all to recognize that the pain, which is the main symptom, pain which can be really severe, is due to sickling. And having said that, it can be just treating the pain, making the blood more fluid, so hydrating the patients, and in some cases transfusing them. This is the standard of care of the crisis. Of course, we want to prevent the crisis, and for this there are many clinical trials ongoing, including treatment for modifying the red cell deformability, for modifying the red cell adhesion, reaching to bone marrow transplantation and gene therapy. Gene therapy will be the future probably, and the first few cases have been already treated.
Right, right. Sounds promising. Further questions from the press? We have plenty of time. Well, thank you again for your presentation. And we move to the last speaker, who had an excellent timing. He came in 2 minutes ago. Dr. John Gribben, our president-elect from Bart's Cancer Institute, London, UK, who will address another EHA topic in focus having to do with CAR T cells and how to make it happen in Europe. John.
Good morning. Thank you very much. Sorry for being late, but I was double booked in another session right before, which just finished. I want to talk to you about our EHA Topics and Focus program in immunotherapy, why we thought that immunotherapy was an issue that should be a topics and focus, and of course what we mean by topics and focus. Each year in the past, Ash, did that again. I'm going to get EHA are going to kill me. Each year in the past,
you are in Stockholm, Sweden.
Stockholm, Sweden. Thank you. Tack. Each year, EHA has had a focus of the meeting, and this year the focus was immunotherapy. Our issue about a topic of the year is that we have it. Next year we have another topic, there's no follow-on, and no one even remembers what last year's topic was. The Topics and Focus program is about setting up a program to tackle issues that we think are of high priority. One, of course, is going to be hemoglobinopathies that you've just heard about. The other is going to be immunotherapy. So let me take you through this. So this is a new EHA program that looks to tackle high-impact issues faced by the medical world today, and I've already said immunotherapy. This year we're focusing on CAR T-cell therapy, and in the hemoglobinopathies we're focusing on sickle cell disease because, of course, you've just heard it's Sickle Cell Disease Day coming up. But we believe that these topics go beyond those particular scope, and immunotherapy is more than just CAR T cells. We view that in Europe there's an immediate need to get up to speed with where the USA is compared to Europe in terms of delivery and being able to deliver CAR T cell therapy, but that the whole field of immunotherapy is going to revolutionize hematologic malignancies, and therefore we're broadening the topic out from just CAR T cells. We're also aware, although that's not my remit to talk about today, the hemoglobinopathies, is that sickle cell disease, of course, is very common, but other hemoglobinopathies are increasingly important in Europe because of the migration issue that we now have, a different population and a different level of expertise available to treat those diseases. So the program's goal, and it's a program, it's not just this meeting, it's not just going to be this year, it's going to be a continuing program that's going to be supported by EHA with a whole variety of other partners, is to raise awareness, provide education, further research, and build a network of experts with the goal of improving the patient care, and in this particular field, increasing access to the therapy, access through our lobbying strategies to lobby with EMA and Brussels to have funding available for these techniques, and access with centres of excellence able to deliver that level, that care to our patients. And of course, through a lot of work that Ton is doing, making these affordable at every level in terms of the price, affordability, and value in terms of what it is we're trying to do. So why immunotherapy? Well, clearly the field's internal speed of change, frontline accomplishments in the medical field, and of course the approval now of agents from two companies in a variety of different hematologic malignancies has created an increasing need for adaptation to new techniques and therapies. And in the first instance, we're focusing on CAR T cells. So in that setting, we are looking to have, as part of this group, and we had, of course, our first session yesterday, our first workshop, but that workshop is the beginning of a program that's going to look at the training requirements, what's required for a site to be able to deliver this type of therapy safely, where safety becomes the major priority. Patient selection, who are the appropriate patients, just as importantly for very expensive therapies, who are the not appropriate patients. Planning for leukapheresis, the multidisciplinary approach of patient management, so the educational needs are not just of physicians, but of scientists, healthcare professionals, our nursing colleagues. Education from the centres that are going to deliver the therapy, but education back to the sites that have referred the patients and will then have to look after potential new complications arising later. Thinking about bridging and lymphodepleting strategies, infusion site logistics, safety, the long-term follow-up, and as already mentioned, a large part we're doing through a whole variety of different approaches through different subcommittees that are all going to work together within this work stream, is thinking about access, reimbursement, mechanisms, and regulatory policy, all of which we believe that we as a large organisation can have a louder voice to be able to advocate on our behalf for these areas, and also to provide information and have partnerships with the patient groups, a caregiver, through education and consideration of quality of life issues. So that, in a nutshell, is what the topics in focus on immunotherapy is meant to be about. To remind you yet again that although we focused this year, we focused in the symposium yesterday on CAR T cells, this is going to include bispecific antibodies, BiTEs, monoclonal antibodies, checkpoint inhibition in combinations. So it's a much broader scope than just CAR T cells. Thank you very much. Any questions?
Yes, questions to Dr. Gribben, please. Yes, in the back. Who are you?
I am Nancy Melville with Medscape Medical News. And regarding the CAR T therapy, are there any measures that are looking promising for bringing the costs down?
Well, of course, the thing that's most promising for bringing the costs down is going to be competition. So the very fact that we've got more than one product is already a step in the right direction. And of course, the scale of uptake. In other words, there are fixed costs in delivering this, divided by the number of patients to whom that's delivered. So if the results continue to be as promising as they look, and in particular, if the ongoing clinical trials looking at bringing this whole technology further forward in terms of where does it fit compared to things like autologous stem cell transplant, and are they more successful if offered earlier, then the number of patients in whom this approach will be offered would rise considerably. And by definition, you've then got the fixed costs divided by the numbers of cases that are being administered. So the cost should come down. It's also the case that at the moment there are still considerable logistical issues in terms of delivering this, in terms of the numbers of sites in which the gene modifications can occur. And those are also ramping up from the different companies that are coming on board, so there should be more resources. And the third one, of course, is through the academic programs that many places have in terms of developing their own approaches to CAR T cells, where it looks as if potentially the costs could also be coming down. So overall, I think it's true that the delivery of the therapy right now is extremely expensive. It's also an extremely expensive niche approach for a small numbers of patients. But we believe that as it broadens out to be more widely applicable, more centres are able to deliver it, more manufacturers and more manufacturing capacity increases, we believe very strongly the price is coming down. And I can tell you that TON is working tirelessly on that very issue, to be looking to see that we can ensure that this, because clearly a hugely important component of access, particularly in Europe compared to the US, is going to relate purely to cost.
Yeah, well...
So it's absolutely something that TON is working on.
And the greatest problem so far is that there are many, many patients on the waiting list.
Yes.
There's a lot of frustration out there. I know in MD Anderson there are a couple of hundred of patients waiting for CAR T cells. So what could we do as a hematology society to promote to speed up the possibilities for patients to be treated?
Well, one thing is that if we can, some of the patients on the MD Anderson list are patients that I've referred because we don't have access to many of the programs in Europe. So as more sites are able to deliver, as the manufacturing capacity increases, and Novartis and Kite, as the examples of the ones who have the products on board, are both working hard to increase their capacity, then we'd have every expectation that the waiting time should come down, and as I've already mentioned, the costs should come down. Now, costs should coming down and costs coming down are two different issues, as I'm sure you're very well aware. But I would view that in Europe, the sorts of prices we were talking about of where CAR T cell costs in the US would be not sustainable within the European reimbursement market, and therefore some accommodation will have to be made in some way for those costs to come down. And we're all aware that that is one of the things that has to happen. How it happens is something I can't quite answer.
Further questions from the press? Yes, please.
I would like to have a comment on where do you see the academic centers, maybe also in production of the cells and delivery, and the industry. You mentioned just the two companies with their production size. On the other hand, I would like to know, do you see a future for academic centers, as it was just in the study from Beijing, that the academic centers develop delivering because the logistics also will decrease, and maybe the time between production of the CAR T cells and infusion may be shortened, so that you, for example, don't have need for therapy in the meantime, and it could be easier if you had academic centers or other delivery centers just here in Europe, if a few of them?
Very good question. I'm not aware of any academic program that has a CAR T-cell program that says we're going to close it down because Kite and Novartis have a product, is the very short answer to your question. There are still many research questions being asked in the CAR T-cell field, and as you're very well aware, there's practically no two clinical trials or two products that are using exactly the same lymphodepleting, bridging and infusion or co-stimulatory domains. So of course, the whole field is exploding beyond purely CD19 as a target to other targets. All of those remain very appropriate academic questions. The whole issue of site registration in Europe then becomes an important one, because we would believe that it will be quite tightly regulated in most of our markets as to which sites are able to deliver CAR T cells. I believe everyone thinks that's a great idea till they find out that their centre is not one of the centres selected, so I think there'll be a lot of competition. And of course, those centres that have an academic programme and therefore experience in delivering CAR T cells are highly likely to be those sites that will be the first among the sites that are registered to be able to deliver this type of therapy. That then leads to, if you like, a competition between what the academic centre is doing and what industry is wanting to do in terms of clinical trial delivery. So the short answer to your question is, I do not believe that the academic programs in CAR T cells are going away. I see them growing. I see them as a very important way of delivering the need we have immediately for our patients. Right now, access through clinical trials is probably the only way that 99.9% of the population in Europe are able to have access to this type of approach right now, even with the products on the market. Most of the managed care markets in Europe are going to be looking very hard about who can and cannot have CAR T cells. And in the short term to medium term, the academic centres are going to be hugely important in filling that gap. In the longer term, those are going to be the centres who will have the most experience, because they'll be the centres that are already delivering. They'll be the centres that are registered to deliver the products which are then licensed. So by definition, you've got almost a self-fulfilling prophecy, is that those centres will then be sustained. And of course, as we already discussed, there's a whole infrastructure. This isn't just about being able to take cells and infuse them. There's a whole infrastructure required to deliver immunotherapy approaches safely. And building that infrastructure in particular sites enables those sites to be the ones that are able to administer this more closely. So I would view more, if the way in which you get to be a site to deliver CAR T cells is to have an academic program, I'd view the academic programs will be increasing and not decreasing.
In addition to that, I know of at least one company who is working on a sort of ready-to-use machinery that can be delivered to each and every institution, so that academic institutions can produce their own CAR T cells. This is one of the developments that's currently ongoing.
Although I've still got the slight concern that producing is one issue, delivering remains. We've been very concerned about the safe delivery of these products.
Right.
And having a site not set up and not equipped to deliver these issues safely is one of the issues that the working group is going to be considering and thinking about. Nobody wants something over-regulated, but this has to be safely delivered.
Right. Okay. If there are no further questions, thank you again, John.
Thank you.
And thanks to all the speakers for their excellent and clear presentation. I guess you are available for interviews now after we close the press briefing. Thank you for participating. Thank you. Pleasure meeting you. Good luck.
Oh, no problem.